In order to mount an effective response to eliminate pathogens or respond to various types of damage, the cells of the innate immune system must first detect their presence. Pathogens possess distinctive molecular patterns known as PAMPs, pathogen-associated molecular patterns, which can bind to specific receptors on innate cells. Additionally, there are DAMPs, damage-associated molecular pattern, which are molecules released by damaged cells and can similarly alert the immune system. The receptors responsible for recognizing PAMPs and DAMPs are termed PRRs, pattern recognition receptors, and can be categorized into 4 groups based on their localization. These include soluble receptors dispersed in body fluids, membrane receptors that stimulate phagocytosis, membrane receptors involved in signaling, leading to the production of inflammatory mediators, and cytosolic receptors. In this video, we will focus on the most crucial class of membrane receptors for signaling, the toll-like receptors. Toll-like receptors, TLRs, are expressed by various innate cellular elements and have the ability to recognize different PAMPs based on their type. Humans have a total of 10 TLR genes, each encoding a receptor capable of recognizing distinct PAMPs. Despite their diversity, TLRs share common characteristics. They are single-pass proteins featuring an extracellular domain composed of 18 to 25 copies of a leucine-rich repeat LRR motif, rich in hydrophobic amino acid leucine. This results in a shape biochemically resembling a horseshoe with a concave and convex part, both capable of binding PAMPs. For receptor signaling to commence, it is essential for 2 TLRs not necessarily identical to dimerize. This dimerization can occur spontaneously between TLRs still free of ligands or can be induced by certain PAMPs. A transmembrane domain, an intracellular domain known as the toll interleukin-1 receptor domain, TIR, which is also present in the cytoplasmic tail of the receptor for interleukin-1 beta. TLRs differ not only with their ligands, but also with their positioning within the cells. TLR1, TLR2, TLR4, TLR5, and TLR6 are situated on the cell membrane, whereas TLR3, TLR7, TLR8, and TLR9 are found within endosomes, binding to any phagocytized PAMPs in these cellular compartments. Now let's delve into the specific ligands for various TLRs and the immune cells where they are predominantly expressed. TLR1, TLR2, and TLR6 are expressed by various cell types, including macrophages, dendritic cells, DCs, basophils, eosinophils, and mast cells. They can recognize diverse components of bacterial cell walls, such as lipomannan, typical of mycobacteria, diacyl lipopeptides, and triacyl lipopeptides, found in gram-negative bacteria, as well as lipoteichoic acids present in gram-positive bacteria. Simultaneously, they also recognize fungal components like beta-glucans and zymosan, a glucan specific to yeasts. Recognition of beta-glucans in particular involves the collaboration between TLR2 and dectin-1, which in turn cooperates with the scavenger receptor CD36 to detect long-chain fatty acids. To initiate signaling, these three receptors create heterodimers, specifically TLR1 and TLR2, and TLR2 and TLR6. This is a unique case among TLRs. All others form homodimers. From a biochemical standpoint, the TLR1 and TLR2 heterodimer recognizes the triacyl lipopeptide due to the presence of binding sites on the convex surfaces of two extracytoplasmic domains for two of the three lipid chains of the triacyl lipopeptide. This process drives TLR1 and TLR2 to dimerize, thereby initiating signaling. A similar mechanism is envisioned for diacyl lipopeptides, which are recognized by the TLR2 and TLR6 heterodimer. TLR4 stands out as the most extensively researched receptor in this category, notable for its unique trait of non-directly recognizing its own ligand. Instead, it employs an immediate mechanism that involves the intervention of other proteins. Let's proceed in order. What serves as the ligand for TLR4? It's a lipopolysaccharide, a constituent of bacterial membranes found in various gram-negative bacteria. Despite variations among different bacteria, LPS maintains a fundamental structure comprising three components: antigen O, core, and lipid A. Antigen O represents the outer section of the molecule, characterized by its polysaccharide nature, and it varies among different strains of the same bacterium. This region, being externally exposed, becomes the target for antibodies. The core, also composed of saccharides, serves as a connection between antigen O and lipid A. Lipid A, on the other hand, comprises a disaccharide to which multiple fatty acid chains are linked. It is precisely these fatty acid chains that play a crucial role in activating TLR4. How does the receptor get activated? In its inactive state, TLR4 contains an accessory molecule, MD2, within its concave surface. MD2 binds to TLR4 from inside the cell. Even before it is exposed, it is crucial for the proper trafficking of the receptor across the membrane. When LPS is present, 5 fatty acids of lipid A bind to MD2, leaving a 6th chain free. This unoccupied chain enables binding to the outer surface of another TLR4, leading to dimerization and the initiation of signaling. MD2, however, is not the sole molecule capable of binding LPS and activating TLR4. In the blood and body fluids, there is also LPS binding protein, LBP, capable of binding freely circulating LPS that has detached from bacteria. Once bound, LBP transfers the LPS to a second protein, CD14, expressed by macrophages, dendritic cells, and neutrophils. CD14 can then activate TLR4 and also serves as a phagocytosis receptor. TLR5 is expressed by macrophages, dendritic cells, and intestinal epithelial cells. It recognizes a highly conserved site of flagellin, the constituent protein of the bacterial flagellum. Concerning TLRs expressed in endosomes, they interact with various ligands more relevant to viruses. which are frequently phagocytosed in endosomes. TLR3 is expressed by macrophages, CDCs, and intestinal epithelial cells. Its ligand is double-stranded RNA, a PAMP present in many viruses but absent in eukaryotic cells, where RNA is typically single-stranded. Poly-1-C, an analogue of double-stranded RNA, is often used to activate this receptor. On the other hand, TLR7 and TLR8 recognize single-stranded RNA. Although single-stranded RNA is also present in eukaryotic cells, it is never found in endosomes, but rather freely in the cytoplasm. Consequently, endosomal single-stranded RNA is likely to be of viral origin. Finally, TLR9 binds unmethylated CpG denucleotides, which are characteristic of the genomes of many bacteria and viruses. In eukaryotic cells, CpG denucleotides, also called CpG islands, attract DNA methyltransferases that add several methyl groups to cytosines. This process is absent in prokaryotic cells. We've explored what TLRs are, their binding mechanisms, and how they activate. Now it's crucial to understand what occurs after their activation. What responses do TLRs induce in immune cells, and how do these responses differ? Let's start by saying that, as is often the case in biology, the activation of a receptor sets off a cascade involving various proteins responsible for the final effect. For TLRs, 4 adapter proteins can bind to TR domains once they've dimerized. MyD88, myeloid differentiation primary response gene 88. MAL, MyD88 adapter-like, also known as TRAP, TR domain containing adapter protein. TRIF, TR domain containing adapter inducing IFN beta. TRAM, TRIF related adapter molecule. The choice of adapter determines the downstream effect. All TLRs can recruit MyD88 except TLR3, which initiates signaling through TRIF. Moreover, TLR1 and TLR2 and TLR2 and TLR6 heterodimers recruit MyD88 and MAL together, leading to MyD88 and MAL signaling. TLR4 recruits MyD88 and MAL, but also TRIF and TRAM. Now let's delve into the specific responses that individual adapters elicit in immune cells. MyD88 is a protein comprising 2 domains. A TR domain capable of interacting with a corresponding domain on the cytoplasmic tail of TLRs, and a death domain, DD. The initiation of signaling through MyD88 and MAL hinges on MAL, which recruits cytosolic MyD88, enabling interaction with the activated TR domains of TLRs. MAL facilitates the formation of the myddosome, a complex involving multiple proteins that allows MyD88 to engage via its death domains with the death domain of IL-1, receptor-associated kinases, IRAK4 and IRAK1. IRAKs serve dual functions. Firstly, they subsequently recruit TRAF6, tumor necrosis factor receptor, associative factor 6, an E3 ubiquitin ligase capable of bridging between an E2 ubiquitin ligase and its substrate, to which a ubiquitin molecule will be attached. In this scenario, the E2 ubiquitin ligase is UBC13, ubiquitin conjugating enzyme 13, working in tandem with its cofactor UV1A, collectively termed TRICA1, TRAF6 regulated IKK activator 1. These ubiquitin ligases effectively attach multiple ubiquitin molecules to TRAF6 by forming K63 bonds, covalent bonds between the lysine K63 of one ubiquitin and the carboxy terminal end of the adjacent one. The resultant polyubiquitin tail serves as a scaffold, providing support for the binding of another protein complex, TAB1, TAB2 and TAC1. TAC1, TGF-beta activated kinase 1, is a kinase that can interact with the polyubiquitin tail through TAB1 and 2, TAC1 binding proteins. At this point, the second function of the previously mentioned IRACT complexes come into play. Being kinases, they phosphorylate TAC1, thereby activating it. It is at this crucial point, right upon the activation of TAC1, that a significant crossroad emerges for the ultimate response of TLR activation. Firstly, TAC1 has the capability to activate specific mitogen-activated protein kinases, MAP kinases, a class of enzymes that trigger various cellular responses. In this scenario, TAC1 can activate C-Jun terminal kinase, JNK, and P38 MAP kinase, which subsequently stimulate AP1, family transcription factors responsible for transcribing genes related to various cytokines. However, in the context of TLR's response, the pivotal role of TAC1 lies in phosphorylating IKK beta. What does this entail? IKK beta is a protein that along IKK alpha and IKK gamma form IKK, IKB kinase complex. Specifically, IKK gamma, commonly known as NEMO, NF-κB essential modulator, binds to the polyubiquitin scaffold, facilitating the interaction between TAC1 and IKK beta, leading to its phosphorylation. Subsequently, phosphorylated IKK beta can phosphorylate IKB, inhibitor of KB, a cytoplasmic protein that under normal conditions binds to the transcription factor NF-κB, nuclear factor kappa, light chain enhancer of activated B cells, preventing its interaction with chromatin. NF-κB comprises two subunits, P50 and P65, which upon release from IKB can translocate into the nucleus and initiate the transcription of various pro-inflammatory cytokines such as interleukin-1 beta, interleukin-6, and TNF-alpha. Hence, this pathway plays a crucial role in activating the immune response in innate immunity cells. However, it has been observed that TLR4 expressed by intestinal epithelial cells, again through MyD88, can stimulate the production of antimicrobial peptides instead of NF-κB activation. This emphasizes the significance of not only the pathway but also the specific cell type under examination. Subsequently, phosphorylated IKK beta can phosphorylate IKB inhibitor of KB, a cytoplasmic protein that under normal conditions binds to the transcription factor NF-κB, nuclear factor kappa light chain enhancer of activated B cells, preventing its interaction with chromatin. As explained earlier, this signaling pathway impacts TLR7, TLR8 and TLR9. In contrast to the previous pathways, it results in the phosphorylation of IRF7, interferon regulatory factor 7, facilitated by IRAK1 and IRAK4. As implied by its name, IRF7 belongs to a family of factors crucial for the synthesis of type 1 interferons, an essential category of cytokines involved in immune activation and antiviral response. It's worth noting IRF5, despite its name, triggers the production of other cytokines, yet always with the pro-inflammatory effect. IRF7 is notably expressed in plasmacytoid dendritic cells. This signaling pathway specifically impacts TLR3. TRIF takes on the role played by IRAK1 and IRAK4 in other signaling pathways by recruiting TRAF3. Just as observed with TRAF6, TRAF3 triggers the formation of a scaffold, bringing together NEMO and TANK, TRAF family member associated NF-κB activator. This scaffold in turn recruits IKK-Y, IKB kinase Y, and TBK1 tank binding kinase 1. Subsequently, TBK1 phosphorylates IRF3 akin to the role played by IRF7, leading to the production of type 1 interferons. In summary, toll-like receptors, TLRs, play a pivotal role in recognizing various pathogens and initiating innate immune responses. Their activation within the expressing cell can lead to diverse outcomes, depending on whether the identified pathogen is a bacterium, resulting in an NF-κB response, or a virus, resulting in an IRF response. It's crucial to note that TLRs are effective only when the pathogen makes contact with the membranes of immune cells, be it cellular or endosomal. Some pathogens, however, can evade detection and propagate directly into the cytoplasm, invading TLR surveillance. To address this, our body has implemented additional recognition mechanisms which we'll explore in other videos.
Thank you for watching. This video was created by School of Biomedical Sciences, Agora. We hope you enjoyed it. If you're curious or have any doubt or questions, please feel free to leave a comment below. If you want to find out more about us or want to support our project, click on the